B8 is all about DNA, and so DNA is, as it says there, store of genetic material, and importantly as well, it's kind of the instructions for making proteins. And so we've got deoxyribonucleic acid. So this is what a nucleotide looks like that makes up our chains of DNA. See, it's made up of 3 sections. This is actually an RNA molecule because it's got this extra OH on there, but the structure is very similar for DNA. So we've got a phosphate charge group here, we've got our pentose sugar here, and we've got a purine or pyrimidine base. So these bases make up the genetic code, and so there's 2 purine bases which look like this, adenine, guanine, and they will also go along with pyrimidine bases, which would be in DNA cytosine and thymine. Now in RNA we replace the thymine with uracil. But for DNA we've got C's, T's, A's and G's. And if we look at making up a chain of these nucleotides, so it's going to form via a condensation reaction, you can see the phosphate group joining on to where that OH was of the nucleotide above it, and it's often referred to as 5' to 3' because if you counted around the carbons 1, 2, 3, 4, 5, you can see we're moving from 3 to 5 like that. And dangling off each of these is our purine or pyrimidine base. Now those bases are able to pair up via some hydrogen bonds, and what we find is that thymine pairs with adenine and cytosine pairs with guanine. It's important to remember those 2 pairings. And you can see there the hydrogen bonds that are involved between those substances. So we've got 2 hydrogen bonds between adenine and thymine, and a slightly stronger attraction between guanine and cytosine. So based on that base pairing, Watson and Crick, work for some other people as well, deduced this double helical structure of DNA. You can see these base pairs in the middle there, and when DNA comes to replicate, it will unwind and a new strand will form using that base pairing to make a replica of the parent strand. So this, you can see the DNA being unwound and then building up a new strand. So that's important where we are replicating cells and we need the genetic information to be the same in the cell that's being produced, and it's also important when we're thinking about passing on genetic information. Protein's also important for, DNA also important for protein synthesis, and I don't think we need to go into too much detail here, but you can see in the diagram all the intricacies of what's happening. And so my DNA is unwound. I've got this gene, there's a definite start codon that's being read, and it's a triplet code, so it reads in 3s. So my messenger RNA copies the gene that we're interested in that's coding for our particular protein. That messenger RNA then heads out into the cytoplasm where it attaches to a ribosome, and transfer RNA molecules again base pair with that, and on each of those transfer RNA molecules we've got an accompanying amino acid depending upon what the code is. And so for our 20 amino acids we've got definite codes for each one, and then those amino acids will join up and we're left with our protein chain. So the DNA stays protected in the nucleus, very important that doesn't get damaged. The messenger RNA heads out and then it can be broken down and used again. DNA transfer, so we can actually move bits of DNA between different organisms, and that's genetic engineering. And so people have been working a lot of genetic engineering with foods, and we might find that we get an entirely new product, we might get a better yield, we might manage to get rid of some unfavorable characteristics. And you can see here some pros of the process and some drawbacks of the process as well.